dear students, I welcome you all for this afternoon session. I already indicated this morning, this afternoon is entirely for bioinformatic analysis, and we have taken the topic primer designing, Sanger's sequence analysis and annotation. And this lecture will be delivered by Dr. Neelam Sheoran. She's a research associate in our division of plant pathology, ICAR-IARI, and she has a thorough, complete experience on genome sequence handling, and she has been working for our whole genome sequencing projects in plant pathology, actively involved in genome sequencing of Magnaporthi oryzae, Magnaporthi griseae, and Ralstonia solanacearum. All the 3 genomes we have recently published in the genome announcement. Presently it is called microbiology resource announcement. And about Neelam, she is a doctoral degree in biotechnology from Singhania University in Rajasthan, and she was involved in a project funded by DBT initially, and subsequently she was handling projects on pathogen genome. So I found her deliberations and her interaction with the student is so important for a project like this. And I'm sure you will enjoy the finer details, what she is going to reveal to all of you. And follow her lecture and also the demo, what she is going to give here with some sequence examples and using some of the softwares what is being used for sequence handling. The highlight of this lecture would be basically on how to handle the softwares. So follow that, and I'm sure the students sitting here and sitting in 2 other universities, you will enjoy this lecture. And if you have any doubts, any clarifications, we'll take up all those questions towards the end of this lecture. So I'll invite Dr. Neelam to moderate this session on genome sequence analysis.
Thank you so much. Okay, welcome you all over here. So first of all, I want to ask you how many of you are actually involved in some type of PCR, sequencing or molecular biology work, whether you are aware of all this terminology or not, so that I can start the things according to that. Right? You all are aware? No. So you are just a fresher to all these things. So the basic thing what we will be starting is the primer designing. So all of you know what is the primer, right? So why do we need the primer? For our PCR amplification. Primer is nothing but it is just a stretch of the oligonucleotide which you require according to your template to make the amplifications, correct? So primers, again we need to have a set of primer, forward primer and reverse primer. Okay. So I'll be telling you few software, those are freely online available. You don't have to purchase it, or no one is the licensed version. So you can use it at your end very easily. One is Primer 3 Plus. You please write it down somewhere, otherwise you will forget. So Primer 3 Plus, this is one of the software, but there are plenty of the software, but I'll be demonstrating using this one software. Okay. But there are plenty of other softwares which you can use to design your primers. You can do it manually also, but since now we have the opportunity to have access to all the software, so that take care of the maximum features or the criteria to design the primers. Okay. So this you write down Primer 3 Plus, and another is Oligo Calculator. After designing the primers, we will be validating whether our primers are good enough to take it further or not, right. That is Oligo Calculator, just to check the properties of the primers which we have designed, right. So let's suppose we have gene A, right. We need to design a set of primer. So our basic requirement is the gene sequence, correct. Either you may be having some reference sequence already with you, or you can download it from NCBI or any other database, or if it is readily available with you, you can use any of the sequence. The purpose is that we want to have a sequence on which we want to design the primers, right, which we want to amplify it further, correct. So let us start that I have a gene sequence of around 1 kb. Okay, first of all I will tell you the main criteria what you should consider while designing the primer. The first criteria is the length of the primer. Length means the bases, the number of the bases. It should be somewhere between 18 to 25 or 30 base pair, not less than that, because that will cause the non-specificity, and not more than that, because then it will be difficult, it will be highly specific then, right. So, the optimum length of the primer, that should be between 18 to 25 bases, correct. Then second criteria is the Tm of that particular temperature, that particular primer. It should be somewhere between 50 to 62 or 65 degrees Celsius, right. Then the third criteria is the GC content of that region which you are picking to design the primer, that should be somewhere between 40% to 60%, right. So, these are the 3 points. Then the fourth point is that avoid the repeat region or the nucleotide. Let us suppose some repeat is there, stretch of T nucleotide or C nucleotide or A nucleotide, you avoid that region. Then the primer, that positioning, it should always be from 5 prime to 3 prime end, right. The designing should be 5 prime to 3 prime. So, towards that 3 prime end, you ensure that there should be some GC is available over there. Why do we need that? Because the AT bonding is 2 and GC is 3. So, that will ensure that your primer is binding tightly on your template, right. So, that also you can take care. Then one more point is that always take care that there should not be any secondary structure within that region. Secondary structure means the sequence should not loop back or should not form any bonding within itself. Okay. Are you getting what I am telling? There should not be any hairpin loop or secondary structure, so that it is difficult for that primer to search the template and sit on that template and start that process, right. So, these are the few features which I am telling you that you have to always consider whether you are using some software. Softwares although take care of all these features, but let us suppose if you are designing it manually, then also you can just look at the sequence and see whether it is GC rich, AT rich. So, there should be the optimum values, what just I told you, these all values are optimum only, right. It is not that the values are this only, it can go beyond that for some specific sequences. Let us suppose you have some sequence that is AT rich or that is GC rich, or there are several repeat regions, then you are left with no other option, right. You have to design the primer over there and you are left, but those cases are rare. But journal, whatever we are designing these days, all genes generally fit into that thing until and unless you have some specific sequence where you want to design your primers, correct. Now, we have two primers, one is the forward primer. Let us suppose this is the whole sequence, this one is the sequence, one is towards the forward and one is towards the reverse, right. So, for each gene you will be designing two primers, the forward primer and the reverse primer, both around 18 to 20 bases each. Okay. So, now you select the sequence whatever it may be, let us suppose it is 1 kb region, okay, and you want to amplify the region somewhere between 800 bases, correct. So, the primer, the binding site of the primer from the forward up to the reverse, it should be 800 base pair. Okay. The software which I will be telling you right now, that is the primer 3 plus software. So, you can pick the sequence if you have already you have some net connection, or you can open the thing primer 3 plus, you can please open it and maybe you can try it simultaneously also. I'll just show you how to do it. So when you Google out this Primer 3 Plus, this is the first window that will come. कुछ connectivity का problem है. कुछ response देर से कर रहा है ना. Actually मुझे लग रहा है दोनों, क्योंकि उसको वो कर रहा है, एक्चुअली इसको करना चाहिए, ये इसका है ना, और वो उधर तक जा रहा है शायद इसका. लेट्स सी अगर इससे होता है तो, या तो उसको लॉक कर दो. अभी तो कर रहा है बट वहाँ नहीं आ रहा है इतना. Pasting your sequence, right? You paste this sequence, then it will ask you what should be the size of your PCR amplicon, right? So let's suppose if you want the whole region to be amplified, that depends on your requirement. But let's suppose you are interested in just 100 or 200 bases out of that whole, then you can specify that thing. There is one option over here, and you can specify that particular region, that okay, I want the region PCR amplicon of around 300 to 400 base pair, right? So once you will click that thing, it will give you a list of the primers based on the criterias which we have just discussed, right? So it will take care of all those things. You don't have to like put your mind that whether it is that or whether it is coming within the range, optimum temperature is there, optimum GC content is there, everything the software will take care, right? The only thing is you will have to pick the primers and send it for designing, for synthesis, okay? प्रॉब्लम हो रहा है ना, कुछ हैंग हो रहा है यहाँ से, नेट का वो ठीक चल रहा है. हाँ अब आया, ये शायद लूज हो जाता है इसका ना, लूज हो गया. अच्छा अच्छा ओके, थैंक यू. Okay, so this is the window, right? Let's suppose I pick any of the sequence. You can download any of the sequence from NCBI, one gene sequence, any of the gene you can download from NCBI. Okay, meanwhile it will get, okay. Let's suppose I'll take this one sequence. Okay, this is one sequence which I copied from, I was already having this sequence. So, I copied this sequence and I have pasted it in this window. Now, go to the journal settings. Now, here it will ask you what should be the product range. So, that should be less than whatever sequence you are putting over here. Let us suppose you are pasting around 1000 bases in that window. So, your PCR amplicon should be less than that. So, it is a basic thing that if you want to design the primer, so the length of that should be less than whatever sequence you are putting it there, right. So, you have to specify over here the different ranges here. You can specify what all you want over there. Let us suppose I will put around 500 to 600 bases, right. So, you specify that thing. Rest thing, if you can just see all the criterias which we have discussed right now, it is already highlighting and it is taking care of all those things, right. So, it is telling you that minimum should be this, maximum should be this, but the optimum should be this, right. So, once you will specify that what should be the size of your PCR amplicon, then you can say, okay, pick primers. So, here another window will appear where it will highlight that where the software has designed the primers for you, right. So, this upper part, the highlighted one, this is your forward primer and this is your reverse primer. Got it? Any confusion? Right. So, the sequence it is highlighting at the top of the sequence, just see this is the forward sequence and this is the reverse sequence. The starting point and the end point of the particular primer, it is highlighting over here. This is the length of the primer, this is the TM of that and this is the GC content of this, correct. So, this is like whenever we are doing all this thing, let us suppose we are blasting some sequence in NCBI. So, whatever hit is the perfect one that it gives at the top. So, same thing is with this, the first one is actually the best out of all the primers, right. Although it will give you another set of primer at the bottom. If you just go down, So, it will give you another primers also, right, with the same sequence, that what is the possibility of designing the primer at different region within the same sequence. So, you have to pick any of one. The first one is always the best, right. So, you just take this, just copy it down somewhere in some Word format or some Excel format, just to have a track that what all are the sequences from a particular gene. So, this is your forward one and this one is your reverse one. Now, it has given you the size, whatever you mentioned, the range you have mentioned is 500 to 600 bases, right. So, it has mentioned that the PCR is 506 base pair. The size of your PCR amplicon, it will be 506 base pair. Any confusion up to now? Any confusion? Yes? Size depends, like what is your requirement. Let us suppose you have a gene of around 1 kb. You are not interested in studying that whole thing, right. You are just interested in seeing the maybe the beginning region or the end region. So, you do not have to amplify that much, like 800 or 900 bases, you do not have to amplify it. You can go with maybe the 100 bases from the forward region or the 100 bases from the, that depends on your requirement. But let us suppose your requirement is the full gene, okay. So, then you need to have the full length, then there you can mention, okay, I want from 900 to 1 kb region, okay. So, that is your requirement only. It is nothing fixed that what I want and what you want, right. So, that depends on my study, that what actually I want to study, whether I want to study some region. Let us suppose I want to check the variability in that whole gene, right. So, there I need to have the sequence of whole gene to check the variability, okay. I need to have all the bases over there. So, there, and if I just want to know that whether this gene is present or absent, then maybe the 200 bases are sufficient just to make it out, because if your PCR will work, if it is, it will give you the positive thing, then it means your gene is present in your particular genome. If it is negative, then it means your gene is absent over there, right. Yes or no, or either if you want to study the yes. See, on NCBI there is an option that you find the gene names are there, right. So, you know your organism. Let us suppose you want to work with any of Bacillus, let us suppose, okay. You know that Bacillus has several housekeeping genes. Suppose you want to go with this DNA 2 gene is there, housekeeping gene. Let us suppose you want to amplify that region, you want to check it in your whatever reference you are taking. So, you simply mention that DNA 2 in Bacillus, it will give you several hits. So, either you must be having some reference organism with you, with which you want to compare your particular sample, the test sample, right. So, you pick that reference, there is maximum, there are it will give you around maybe 50 to 60 hit in a single go. So, you pick that particular organism that belongs to your study, okay. Let us suppose it is some Bacillus megaterium or Pseudomonas putida, and you want to, your organism is that. So, obviously, you will be picking the putida only or the megaterium only. So, that depends, okay. Let us suppose if it is some, we know that we have some sample, suppose human sample is there. I do not have any other entry in database pertaining to the human. What is the nearest one? Maybe I can go with the monkey sample, right. So, the nearest thing in the evolutionary phase, you can pick that one also, okay. That I'll be coming to that. This self and any is the point which we discussed, whether it forms any secondary structure, whether it forms any self loop, self dimerization is there. So, these are regarding the structure of that particular primer, okay. Whether it is annealing among itself, because let us suppose I have a sequence where 3 C are there in the beginning, 20 bases are there, there are 3 or 4 C's in the beginning, and maybe 4G towards the end or the middle. So, what will happen, since both are complementary, it will just bend and form the hairpin loop type of structure. Got it? So, it will give you these two primer sequences. This is the forward one and this is the reverse one. Another thing, this mistake people generally make in the beginning, the forward primer sequence is actually the sequence of the gene which you are putting it over here. But the reverse primer, the sequence of the reverse primer is reverse complementary to the gene sequence. Please make this point in your notebooks that the reverse primer will be the reverse complementary of your gene sequence. Then only, because company people will ask you that what is the 5 prime to 3 prime sequence that you want to synthesize. What will happen, let us suppose you have a template, you know the things are double stranded only, right. This is the double stranded. Okay. So, your primer is designed, forward is somewhere here, reverse is somewhere here, let us suppose. So, your both the strands are in reverse complementary, right. Both the strands are reverse complementary of each other, correct. One is 5 prime to 3 prime, another is 3 prime to 5 prime. So, the primer should also be in that orientation only. Can I have one? Okay, I will just show you how it works. Let us suppose this is your sequence, this is your double stranded sequence, right. Everybody can see it. So, this is your 5 prime to 3 prime and this is your 3 prime to 5 prime. This is how we have our the genetic material with us, right. So, this is your forward primer, this is your reverse primer. So, the forward primer, the orientation will remain the same whatever the gene sequence you are picking, right. But the reverse primer, if you can just see it over here, the primer starts with GGGT, right. If you go down and you try to match that sequence with this, you will see that it is not matching. Why it is not matching? Because that sequence is actually the reverse complement of your gene sequence. Any confusion? So, if you read this particular sequence in the reverse orientation and in the complementary orientation, you will just see GG, this is CCCA, right, and at the top what we read is GGGT. So, the software is already converting this sequence into the reverse complement. So, it is making it ready made for us that okay, you pick the sequence and send it for synthesis, correct. Any confusion? Please, you just take care that the reverse primer, one primer is always the reverse complement of your sequence. Do not give it like that, like the forward primer is this one, reverse is not this one, reverse is the reverse complementary of this one, okay. Otherwise what will happen na, once the primer anyway will bind at your sequence, but it will amplify the region beyond that, like it will start amplifying the region from here, which you do not want, okay. So, now the thing is, the primers are already with you. One more point that is very important, that the TM difference of your forward primer and reverse primer should not be more than 5 degrees Celsius. Why this is so? Because you need that your primer should bind, you are putting everything in a common pool. So, your binding should be for both the primers at a single temperature, because annealing temperature you will be putting just one annealing temperature, okay. And your annealing temperature, that should always be 5 degree less than your TM, the melting temperature of this primers. Can I proceed further? Okay. So, now because you don't have to do much thing, primer is already with you. Okay, you had entered your sequence, software has given you a set of primer. Let's suppose there is some problem, or now we will go with for the validation of these primer. So for validation you can write down one more software is there, that is oligo calculator. You can write it down. So after I will get those primers, what I will do, I will validate those primers whether some complementary structure within that primer it is getting affected, it is affecting that our primer sequence, and which will further affect our PCR. So this sequence I will paste it over here, and then there is one option, check self complementarity. So you can just have a look. There are the steps which we just discussed, that there should not be any hairpin formation. So if you will just see this, it is saying it is none for this particular primer. Okay? So it is free from any hairpin formation. Then 3' complementarity, that is also none. Okay? Then some self annealing sites, that is also none. It means our primer, whichever the software prepared for us, that is good enough to take it further. Otherwise, if it would have highlighted some region that was making some secondary structure, or the internal bonding is being formed, then we had to avoid that primer. So in that case, what we can do, wherever in that particular, let us suppose 4 bases at the end, those are problematic, those are forming some bonding with the internal part. So we can avoid those 4 bases from that end, and we can take the 4 bases from other end. So that we can do it manually also. So it's okay. Can I proceed further? Right. So the same activity you have to do with both of your primers, with the forward one and with the reverse one. So if some problem, if it will highlight something that okay, some hairpin formation is there, then you please do not take that primer, because that may not work, and then you will be struggling in optimizing your PCR reactions. So any question regarding this, regarding primer designing, any doubts please, otherwise then I will go ahead with the sequence analysis. So the company people will be asking you, so then what you can do, you can label your primer. Label means, let us suppose I have the gene sequence was DNA2, okay. So how I can label, DNA2 dash forward, DNA2 dash reverse. The orientation should be 5' to 3' for both the sequences. You send it to the company people, those who are synthesizing the primers for you. They will give you the primer in particular concentration, some nanomolar concentration they will be providing you. Generally they provide the lyophilized form of your oligonucleotides, right. So they will deliver it to you, then you resuspend according to the concentration which the company will be providing. The stock solution, always make it the 100 picomole per microliter. Then whenever required, then you dilute that primer to the required dilution, maybe 10 picomole you want or 5 picomole you want. You do the dilution as and when you want, right. And that stock immediately you please freeze it in -20, because primers are very prone to degradation, because this is a small single nucleotide only, right. So if you will do a lot of freeze thawing, then the last nucleotide that has tendency that one by one it start degrading, and that effect will actually play a key role in your sequencing, because the sequencing data generated from the degraded primer, that is always a noisy or a mixed data, right. So always handle such like nucleotides or PCR amplicons or whatever you are handling, because these are very sensitive at room temperature or at higher temperature. So always try that you keep it storing in -20. Whenever required, you just thaw it, make a required volume of your like 10 picomole or 5 picomole, and then use it in your PCR. Okay. So, can we proceed further? Right. So, this is a one step that how we have to design our primers for PCR amplification. So, now PCR, what all you want? You want the template which you are already having, right. Primers you have already designed, forward and reverse primers are with you, DNTPs you want, enzyme you want, and buffer you want. So, you can select any of the companies for that. Many companies provide the cocktails also, or you can individually do it also, right. Then the annealing temperature, whatever we discussed right now, if you will just see the temperature the mentioned over here, now it is 59 over here and 60 over here. So, this PCR optimization you can do somewhere at around 55 degree, it will work, definitely it will work, right. It should be 3-4 degree less than that. Okay. If let us suppose you are getting some non-specificity, if your primer is, okay, once you are through with your primer designing part, always try to do, there is one primer blast, NCBI primer blast. What it will do the blast analysis for your stretch of oligonucleotide, and it will give you the hit that whether it is amplifying in other genomes also. Okay. So, if you want a specific primer, then you please do the NCBI blast also. That will highlight that in because suppose you have a sample that is unknown to you, you do not know what it is, right. So, it may be anything. So, from there you can have an idea that it can bind in other genomes, other organisms also. And one more thing, let us suppose you have some organism, the primer designing should maximum be from the conserved region. Conserved region means, let us suppose, because there is a variability like 1, 2, 3, 4 basis variability is there among, because that depends on organism to organism how variable it is from each other. Conserved region means all the bases at that particular area, those should be similar among a number of a set of organism. So, for that thing how we proceed further, let us suppose I want to design a primer for some gene in particular organism, but within the organism also there are variabilities. So, what I will do, I will just do the NCBI blast of the sequence which I am having. It will give a number of hits, maybe around 100% similarity, 99% similarity. I will pick around 10 to 15 sequences from NCBI blast, download the sequence, do the multiple alignment of those sequences. Multiple alignment using any of like Cluster W or any other multiple alignment software you can use. So, once you will multiple align it, then you will come to know that the region that is conserved among all the sequence, you please try to design the primer in that particular region. Because if you will pick the variable region, then your primer will be specific to that particular region only. Okay. If your organism is that only, then only it will bind and amplify the things. But let us suppose if variability is there, then your primer will not bind and proceed further. Okay. So, the primer designing should be primarily from the conserved region. Okay. No, you can manually also you can do, but you can put that region, you can put that region, because once you will, let us suppose you will be picking from the blast whatever you did for your particular sequence, you did the blast, right. So, around 10-15 sequences you picked from the blast hit, okay, that table you had picked, you download those sequence. Now, when you are multiple aligning those things, you will pick the region which is actually similar among all, okay, then you can design the thing. That you have to cross check. Now, after the primer has been designed, then you have to cross check whether your primer actually lies within that conserved region or not, right. So, that you have to verify. That is your validation part only, that whether you are designing the primer at the correct location, or if some variability is there, then you can face problem in the PCR. Okay. Because as we are doing with like I do not know how many of you are handling some bacterial or some fungal sample, or maybe I do not know much about that virus, that how it happens, but let us suppose you must have studied the 16S rDNA amplification, right. So, that is basically for the bacterial identification. For fungal identification, we go with this ITS identification, right. So, those primers which we are using, those universal primers, although we want to check the variability among the organism, then only we can make it out which bacteria it is or which fungus it is. But the primer designing, the primers which we pick, those are the universal primer, because that sequence is actually conserved among the bacterial species, the bacterial genus, and the species it will give based on the variation in that particular amplicon, correct? Any other questions? Yes.
In primer glass, you have to blast the primer separately forward and
Yeah, you do it separately.
And there are certain values upstream here. Can you please open the primer?
Here it will take time. Actually, net is taking time. I can come to you and just see.
It's like some these things are there, so we need to put something in those also, or we need to just put our
Just primer sequence, just primer sequence you put.
and
Forward
nothing
sequence.
on forward, only forward.
Forward
sequence.
only you put, and you just go down and do the blast.
Blast. It is get primer
No,
sequence.
no, not this one. It's a NCBI primer blast.
Yeah, that primer blast we have opened only.
No, maybe you have clicked some more thing also over there.
No, it's like.
Okay, that I'll show you there.
Okay.
I'll continue this one, and that I'll show you there.
Okay.
Right? Okay, now your PCR amplification is done. You can send those PCR for the sequencing, for the Sanger sequencing. You have to send the amplicons, and of course, you have to send your primers to the company, because they will be using these primers for the sequencing purpose. Now, that depends like how much region you want to get it sequenced. For Sanger sequences, company people, they commit that they can give you around 500 to 600 good quality bases, right. But let us suppose if you want to go beyond that, then you have to go with the multiple primers. Multiple primers means, let us suppose if it is around 1.5 kb region you want to sequence. So, you have to design one internal primer also, so that the whole region, one is from forward, another is from reverse, and one internal primer also you can design for the sequencing purpose, not for the PCR purpose. PCR you can amplify up to 10 kb also, that is not a problem, but sequencing, Sanger sequencing, you have to go a fragment wise, right, 500 to 600 bases per read. Per read here I mean that with a single primer, the sequencing company can generate only 5 to 700 good quality bases. Okay. Because the sequence you are already having with you, right. The sequence, let us suppose you have around 1 kb or 1.5 kb, the sequence is with you. Forward and reverse primer you have already designed to amplify that region. Now, some internal primer you can design in any of the orientation, so that the company people can use that and sequence that region. No, manually from this software only you can use. Because here you will be putting that sequence. If some internal primer is there, then the location you can find it out. Here it is highlighting you that where it is designing the primer, right. It is highlighting. Let us suppose I would have entered around 900 to 1000 bases. So, one primer at this location, another primer at this location, then this must be the internal primer only, right. So, the location of the sequence, that you can decide, that at which point you can design one more primer. There are two things, either you design one internal primer, or what you can do, let us suppose you have already designed one primer here and one primer here. So, there you can design a two set of primer, like this is F1, okay. Somewhere here you can design R1, okay, F2, and this is R2. So, what will happen, from this you will get one sequence, and from this you will get another sequence. If you will align them, or if you will make a contigue of it, then you can get the whole sequence out of it, right. So, overlapping sequence you can take, and then the contigue can be formed, okay. So, how many of you have already done some Sanger sequencing? Anybody did? Okay, you all did. Okay, what is the Sanger sequencing then? We will not go too deep. I will just tell you that this particular samples which we are generating in day to day in our lab, we are sending to the company people that we want the Sanger sequencing of this. So, we want to give them the PCR product, either purified or unpurified. If it is unpurified, then the company people will purify, or if we are sending the purified only, then the company people will sequence it using our primers, whatever we are giving them, right. I will send the PCR amplicon, let us suppose 100 kb product I am sending it to them. I will send a specific concentration of primer to the company people, that you please sequence this region using our primers, whatever we are sending you. So, after sequencing, they will be giving you data in 2-3 forms, right. So, one form is, which we generally think that okay, the FASTA files are, we actually require the FASTA file, the sequence ACGT, whatever it gives. The another thing is that AB1 files, that these are the chromatogram files that are directly generated from the instrument. Those are actually the raw files, okay. So, we cannot do the manipulation in those files. I will just show you what all are the formats. I have some sequencing data with me, just a demo. So, this is the data that the company people will be providing you, right. Each sequence means one data will be from your forward primer, another will be from your reverse primer, right. So, let us suppose this is your forward one and this is your reverse. I just told you the AB1 file, this is that AB1 file format, okay. There is one more software you can write down, there are plenty, but you can write down one, Finch TV software, Finch TV, FINCH TV. That software will help you to view this chromatogram files. There is another software, DNA Baser. So, what will happen, this will be your chromatogram file. When you will click it, then you can open with any of the software that you have already installed in your computers, right. If it is Finch TV, then you can open it with Finch TV. If it is something else, you can open with that, right. So, this is the AB1 file, the chromatogram file, which I will be showing it to you. Then the another is FASTA file. So, this will be your FASTA file. The company people will say that okay, this is the FASTA format of the sequence, this was your sample name, this was your primer name, PGK forward, okay, and this is the sequence which the company is providing. But never ever go forward for the analysis purpose with this FASTA format, because they are saying that this is CGT, but how good that particular base is, you do not know, right. So, for that thing you want to know the quality value of each base, right. So, the chromatogram file will actually tell you the quality value of each and every base with from like if the Q value is more than QV value, right. So, Q value is more than 30, then you can consider that base, otherwise there is some doubt that either the sequencing data is not good, or if something is missing, or the machine condition is not good, or there is some impurity in your sample, it means that sequencing data is not good at that point, right. So, never ever just take that this thing and go ahead ki ha mera data aa gaya and now I can use this data, because there are lots of error in this particular like this ACGT, there will be lots and lots of error over here, right. If the data is mixed, then also the machine will pick any of the base, either A, C, G, T, right. Machine will do its work, because it is getting all those dyes from the machine to pick a certain, it has to pick a certain base, because the machine's protocol is like that. Either it is A, G, C, T, if let us suppose 2-3 peaks are there, then also whatever is higher it will pick, right. But the problem is that whether that base is a clear base or it is mixed with other bases also, right. So, that will have to make clear before taking the sequence further for analysis. So, the Finch TV file which I had just shown you, this is the Finch TV. So, your data, this is the chromatogram file of each and every base of your sequence, correct. So, if you will just go down, just scroll it little bit, you will get the whole sequence. This is the data which I am showing you, very good data. What will happen with the bad data? Either you will not get any of the peak, or one peak is overlapped by another 2-3 peaks, right, or the peaks are not properly resolved. Why I told you in the beginning that company people, they commit only 500 to 600 good bases, because what happens in the instrument, after 500 to 600 bases, the peak resolution that declines, okay. The clear cut, now you can see the clear peaks, right. In the beginning, if you have seen, just see the beginning, you have seen the clear peaks, right. Just see, those are very like crisp peaks are there. But if you just go forward and go beyond that, can you see the resolution of peak that starts declining after around 600 to 700 bases. So, these and if okay, that quality value which we were just talking about, on the top of each base, let us suppose this C is the base, can you see a bar diagram? This bar can you see on the top of this C? This is the quality value, okay. So, if you will go with that FASTA format, you will assume that okay, C is there, but how good that C is, whether that C is genuine or not, that you have to go to each base and check. Now, if you go down and check the Q value, here is the Q value is 53, that is more than 25. So, the peak is authentic peak, right. But if I pick this one, let us suppose this one, this C I pick, can you just see the quality value? Now, the Q is 9, and you just see the quality of the peak, although it is recognizing it as a C, but there are some overlapping peaks also, those are quenching the quality of this particular C. So, I cannot rely on such data. That is the reason the region around 500 to 600 bases, the good quality value is there, that region you consider. Beyond that, you need to get it sequenced with another primer, right. So, this is one sequence which I will be, although the sequence will be read up to 1000, 1200 bases also, but the quality is being compromised. So, that is the reason we always try to amplify, to sequence the amplicon from both the sides. Why from both the sides? Generally we call it as a X coverage. I think you might have heard about it. X coverage is like how many times you are reading a particular base to confirm that that base is actually that base. If it is, let us suppose, if I am reading something ACGT once, now whether G is G, I do not know, but if I'll sequence it 2-3 times, every time it is coming G, 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 then I can be sure that it is not erratic, it is actually the GP. So that is the X coverage, means how many times you are sequencing a particular base to be confirmed that that particular base is this only. So if you are getting the sequencing done from both the forward and the reverse primer, it means you are sequencing a particular area 2 times. Let's suppose there is some problem in one sequence, right? So that will be taken care by the second sequence. So the many times you will be sequencing a particular region, more confirm you will be. Okay, because I'll not go in deep like for if the high end sequencing, this NGS and all, they are sequencing it 100x, 1000x, just to make confirm, because they want to clear off the error, that there should not be any error in that particular sequence. So more deep you go, more times you will be sequencing it, more times you are getting it confirmed that this particular base is, yes, this is the base, correct. So what we will be doing, let's suppose I am amplifying around 500 bases, I will be giving it for the sequencing. So 500 bases the company will be amplifying 2 times, one from the forward, one from the reverse primer, right. So I will be sure, let's suppose if problem is there, suppose this is this C I am not able to confirm, or this G, or maybe this A I am not able to confirm whether it is A or not, then I can go back in the reverse sequence, this is the forward one, I can go back in the reverse sequence and check this region whether this G is having a good quality. If that G is having a good quality, then yes, I can consider this G for my analysis purpose, right. So this is a very tricky thing, and you have to actually spend a lot of time in doing such analysis. So always generate a good PCR amplicon, your primer should be free from any degradation, the company should provide you the clear cut peaks, it should not be something messy कि कुछ नीचे नीचे कुछ और peaks आ रही हैं, if some impurities are there, if the sample is not been purified properly, then the DNTPs and DDNTPs which you will be adding at the time of your PCR and sequencing, that will be actually affecting the quality of your main peak, right. So that is the reason we do the purification of our PCR, correct. So the I'll be provided the 2 types of sequence, one from the forward and another for the reverse, right. There are many other formats also which company will be providing, but that is of not our use. So we just need the chromatogram file and the FASTA file. FASTA file we will consider, but we will go with the chromatogram file first. If there are some errors, we will try to check those errors, and if that is actually the correct base, then we can take it further, right. So here there is one option, I information, here also that same FASTA the sequence will be there, details will be there, like it will highlight you each and everything, that like how many A's are there, how many, just go there, how many A are there, C are there, G is there, T is there, right, in this whole sequence. So this is how your data will look like from the company. Whenever you will get the data, the very first thing is you just open the chromatogram, check the graph, check these peaks. If these are good peaks, then you please take it further, otherwise just dump it into the dustbin. That is of no use. Don't go with the FASTA files only, that okay, ACGTs, they will give you something, but don't go with that, right. So now the second part is, you got both the data from the forward and reverse. Now what we discussed is, like forward is from here, the forward has been designed, here the reverse has been designed, right. So the company will give you around 500 to 600 bases from this and 500, 600 bases from this, okay. Now the second step will be to join both the sequences, right. What that joining will be called as? Contigue. So, we will be making contig using our forward and reverse sequence. What will happen? The overlapping region it will consider as a 1 and it will join like 2 fragments, it will join and make it a single fragment. Then this whole thing will be 1 kb region. Okay? Any confusions? So, the second step will be the contig formation. Once you are clear with the quality value of your sequence data, second step will be the contig formation. That will be that can be done using the DNA baser also. That trial version is freely online available, but you can purchase the licensed version also. One freely available more is there, that is a BioEdit, that also you can write down. BioEdit software that you can download and use it offline. I will just show you how to import those files, and there are other softwares also. One CLC is also there. There are plenty of software for all these applications, right? So, I will just open one file for you. This is once you will download that BioEdit, then this window will appear. You can use it offline. Let's suppose you have downloaded it, you got the sequencing data, you don't need to have the internet connection and all, you can make the contigs as and when required. So, this window will appear, go to file, you can write down this thing and maybe after going back to your hostels you can practice, otherwise you will forget whatever, because these things are new to you and you may not be comfortable in handling it alone. Go to new, then file, then import, and always the software which we are using, the softwares will be asking for AB1 files only, because that is the actual file that is generated through the instrument. ACGT you can write down any sequence, right, but this sequence you cannot change. The peak color that will recognize the base, what base is this, like A is this, black peak is this, then this is G or C is blue. So, 4 dyes have been used for sequencing this thing, each for each base, right. So, there is one option that it will ask you that you import the file, you just go and I will select the region. I will just add on these AB1 files. Both from down you can select AB1 files only, so it will highlight AB1 only, right. So, now it has imported both the sequences. One is the forward one and another is the reverse one. Can you just see from here? This is the forward and this is using the reverse one. Now, go to accessory application, then there is one option CAP contig assembly program. Click on that, run application, just close this window and it will give you one. Now, this is the forward one, this is the reverse one and this is the contig. If you just scroll towards the left, you can just see there is the region that only forward is having and there is the region that only reverse is having, and this, can you just see this extra region, because the central region will be overlapping in both the sequence. So, it will consider it as a 1, but there must be some sequence that is not present in either of the thing, either it is present in forward only or because you are not sequencing it completely 2x. If it is 2x, if it would have been a 500 bases only, then you would have got the complete 2x coverage. All bases will be overlapping, right. So, this sequence is required for your for the work. Now what you can do, you can copy the contig and paste it somewhere in your some Word file you can make with your gene name and your primer name, right. Let's suppose I will just copy it and paste it somewhere. I will just show you how I did it. So this file I am showing it to you, where I have already this contigs, can you see? So this whole thing is actually the sequence of both forward and reverse. I made the contig and pasted it in some Word or notepad document, whatever it may be, right. So now once your this file is ready, now the second thing is that how good the bases are. How good the bases are means it will be giving you from the beginning up to the end. But if you will go to the Finch TV and just see the sequence, not all the bases are of good quality. You just see the beginning around 30 to 40 bases you generally miss out in your Sanger sequencing. That is mandatory, right. So what you can do, let's suppose you are amplifying some region from 1 to 500 bases, right. But initial 30 to 40 bases you will definitely miss out in Sanger sequencing, because the reaction starts, the resolution is not proper, but once the machine is on and it will, that is the chemistry which they are using, is that is not supporting the initial bases. So you always consider, let's suppose your requirement is initial 10 bases only, suppose, and you don't want to miss any base from your sequence, then what you have to do, the primer designing, you have to do 50 bases upstream of that region. Got it? 50 bases upstream of that region, because that depends on your requirement. If you don't want to miss out anything, then you please, the primer designing should be done around 50 to 100 base pair towards like 5' end and 100 to 200 base pair towards 3' end. Then only you can get the whole sequence whatever you require. Clear this thing? Any doubt? उसको पूरे को सेलेक्ट करके, वो इसमें माउस नहीं चल रहा है, उसको सेलेक्ट करते हो कंट्रोल सी करके, जब आप कंट्रोल वी करोगे ना वर्ड में जाके तो वो हो जाएगा, हो जाता है वो। ये मेरे माउस से नहीं हो रहा है यहाँ पे ना, नहीं हो रहा है यहाँ पे, आप लोगों का नहीं हो रहा? अच्छा मैं बता दूंगी, हो जाता है, ये इजीली हो जाता है, इसमें कुछ नहीं। Any doubts up to here? Can I move further? Okay, now once the contig has been formed, you have already saved that contig with you. Now you want to verify that all bases should be good enough. Suppose I had somewhere like, okay, I'll make one contig. What I'll do, I'll try to pick some region. Okay, one more thing. Let's suppose at some point the data is not good. Instead of we have forward and reverse sequence. In one sequence, it is reading, the software is reading as C, but in another reverse one, it is reading as A, suppose. But I want to confirm whether C or A. Contig will also give it some base, whether C or A. But whether I have to go with that or not, that I have to go back to my chromatogram file and check that particular base. If the quality value of that base is more than 20 or 25, then I can, let's suppose it has misread that C as A in the reverse sequence. Suppose these two sequences I had taken this forward and this reverse. Okay. Here it is reading C, but here since the reaction is dying out towards the end and the resolution is not proper, the quality is not proper, the software has read it as A. Okay. BioEdit will read it as C or A. That also depends, like if you will use the licensed software, that will confirmly give you the correct base, but if you are using some online software, then definitely some errors will be there that you have to check manually. Okay. So, if this is C and this is A at this particular base, I need to check. What I will do, I will copy this sequence, go back to the chromatogram file, paste it here in this window, like just now I had pasted, and then find that particular sequence. Next to that particular sequence, that C will be there. Okay. I will check the quality value of that C. If this is good peak, then I will edit it as C. I will not consider A, but I will consider it as a C. Okay. So, how this happens, there is one another software that is CLC sequence viewer. CLC sequence viewer. Okay, I will just give you one example how you can do it. Let us suppose you have one reference sequence with you, okay, some gene sequences with you, and you had amplified, you had around 10 samples. You had amplified using a particular set of primer. So, now you will be having those 10 sequences with you, okay, along with your reference. Total 11 sequences will be with you. Okay. So, for 10 sequences you will be making contig, like I just told you. Make the contig, put it in a single file with the FASTA format. FASTA format sample 1 and let us suppose gene A. FASTA format sample 2 gene B. 1, 2, 3, 4, just one after another you paste the sequence, convert it to text file. Okay. Then text file you can import over here in this CLC software. See, it has several applications. You can use it for making alignment, you can use it for converting a sequence to the protein form, you can use it to make it reverse complement. There are several options, you can explore it if you just go to toolbox, alignment you can do, then trees you can form, the phylogenetic trees, that also you can form with this. These are the how you can join the sequence, how you can convert the DNA to RNA or DNA to nucleotide to protein. So, there are several applications, you can explore it later, right. So, what you can do, once you are ready, let's suppose I had made one file, I am having this file for contig, right. I already converted it into text file. Now, I will import this text file into that software to do the multiple alignment. What is the purpose? Because I had sequenced around 10 of my sample using the same gene. Okay. I want to compare with each other. The sequence should be same in all the samples. It should be like in sequencing, some bases should be more in the beginning, some will start sequence earlier, some may start at a later stage. So, my purpose is to get a clear cut basis, 1 to 500 should be same among all. Okay. So, what I will do, the raw sequence I will make the contig, make one file, word file, convert it to text file, import it in this software. So now I'll just show you how to import it. Here one option is there, file, import. Then I'll pick that file which I already made, this file I made, next, next. I think this computer, this version it is not supporting, but I'll show you whatever is already having na, in this software. Now this file, earlier only I had imported some file over here. If I'll import that file, this format will come, this format. Okay, the red icon, na, red bars will be coming. So for making the multiple alignment, I'll select this, go to this, and create alignment. Okay. Now this has been already been selected. Whichever each gene, one file will be generated, right? You cannot do it among all the genes because sequence will be different. Alignment you can do within the similar thing only, na. If it is A to A, then only you can align. A to C you cannot align, right? So you can go ahead. Next, this will ask you very accurate. We will select finish. So now you can see at down it is making the alignment. So your files will be aligned somewhat like this, right? Because at some point data may not be good in some sequence, it may not read it correctly, it may not align. That is the reason it is showing some gaps somewhere, right? So there is not a uniform data in among all these 4 sequences. Now these are the 4 samples, these all are the 4 samples and with one gene. Okay, so 4 samples data I have imported within a single text file. If you just go down, after certain days the data will start appearing uniform. Can you just see over here? Here some uniformity is there, right? Everywhere it is C, everywhere it is G. If you go there, then again it is not matching. You just go further down. If you can see in 2 of your sample, you are getting CC. In another 2, you are getting GG. So that variability is there. But again, now the question is whether this C or G, these are actually different or it is some like misread. So for that purpose, you will have to go back to your FASTA, those chromatogram files and verify. One by one you have to do. What you will be doing? In sample number 1 file, this is sample 1, 2 and this is K and A. So what you will be doing? You just select the sequence, right? By right click you can copy it, go to Finch TV. Actually this is some other file, so it may not highlight over here. So paste it over there, then say enter. Okay, pattern not found because this is something else. Otherwise if I would have tried with the same sample chromatogram, let's suppose I'll do it just for your clarity, I'll just make it once. Let's suppose this is the sequence which I had copied from that CLC software. Okay, copy, paste. Correct? Now what is the next base to the selected sequence, it is T only, right. Although the quality is not very good, because you just see there may be some resolution issue over here. Just go down and see, there may be some, you just see, whenever there are some multiple nucleotides are present in the sequence, then the resolution of those peaks that decreases. Okay. So, the machine cannot read it properly what base it is. The quality of that particular base that quench down if the resolution is not proper. So, here you can find there is a stretch of CCCC. So, there is because we are using some enzyme for sequencing also. So, there is a slippage of that tag that this thing appears because of that, right. But if you will just see the peak is quite good. So, if there in that particular this alignment, if this T is there, then I will go that okay, this T is actually T, I do not have to change it. But let us suppose if this T is not T over here, I will check in both the sequences, in the forward also and in the reverse also. If in forward the quality value is good for T, and in the reverse the quality value is not good for A, T to A I am considering. If the A is not good quality value and T is with a good quality value, then I will go back here, I will select this particular base, right click it and rename the sequence, right. Okay, I will just edit this, select, edit selection. Let us suppose I want to change this C to G. Okay. Okay. Now what happened? That C has been converted to G. But this editing you can do only after verifying from the quality of your chromatogram. Do not do the editing without any confirmation, otherwise you will spoil your sequence, right. So, this form is called, first was the Contig formation and now this is the editing of your sequence, correct. So, this is the example which I am showing you how you can change the base if the quality of that particular base is not good in one read, but it is good in another, then you can replace it. But if this G is actually G in both forward and reverse, then you do not have to change it. Then that is the actual variation in the sample, that is the actual variation in your sequence, in your genome. Okay, then do not change it. You can change only if it is a sequencing related error, process related error. If it is actually the good base in both the sequence, then please do not change this base. Do not do any unconfirmed editing, correct. So, this is one step of editing. Another step is while we were doing the multiple alignment. So, what we do, although here too many unsimilar things are there, otherwise in the beginning it generally do not match, at the end it generally do not match, because one sequence will start at one point, but maybe another sequence will start 10 bases downstream to that, right, but we want to make it a uniform. So, that step is called the trimming of 5' and the 3' end. So, I will just show you how to do it. Let us suppose I consider that, can you just see this sequence, it started bit early, but another 3 sequence that are starting at a later stage. So, it is missing around 8 to 10 bases from each, and others are also not similar. So, because the beginning I told you na, that beginning and end the data is being compromised. So, we can trim that region to avoid any confusion what is that base, because if I will be sequencing around 600 bases, so I will consider that okay, at the end around 500 will be in my hand. I will chop off another like 100 to 200 bases from both the end. So, whenever you are designing the primers, you consider what actual sequence you want. You always design the primers 200 base pair more than the region whatever you required. Correct? So, what I will do, I will just select the sequence. Suppose here to here I selected. Okay. From now T it is getting uniform, for example, right. So, now this T it is uniform. So, what I will do, I will select all the sequence, right click, delete selection. So, now what happened? The non-uniformity that actually cleared. Now, all bases are starting from a one point, right. So, same thing happens in the 3' region also. Can you just see, after a certain length, I think only 2 samples are getting sequenced, but in others the sequencing stopped, right. So, what I will do, I will again select this thing, the whole thing, right click, delete selection, right. So, now what happened? Now, all the 4 sequences are being normalized. It starts from 1 and it ends at the same point, right. Then only I can compare if all the 4 are having the similar number of base. This is the trimming part, 5' to 3', and in between if some variation is there, let us suppose you can find some gap is there, this is gap, okay. If I will just see it, I will assume that maybe it is a deletion, right. It is a deletion of that particular base in my sample, but whether it is actually deletion or not, that I will have to go back to my chromatogram file and check whether after this A again C is coming with a good peak, or some confused or some blurred or some mixed A is also coming. If something is there after that, then I have to consider in both forward and reverse data, and then only I can replace this deletion with the A, correct. Then this deletion is not actually the deletion, this is the bad quality sequencing data which is affecting over here, correct. Am I clear? So, now you are through with the trimming, you are through with the editing according to the chromatogram file. Please do not do the editing blindly, otherwise you will get confused कि कौन सा base delete किया था, because all seems to be similar only. So, please do not do that. So, after this, the file which I will be showing you, this is the another file that is already been trimmed and checked, each basis has been verified using those chromatogram file. So, now these are the ready to use file to submit to the GenBank, correct. Any confusion up to now? Yeah, you want to say something? Okay. So, now here if you can see, it everything is starting from the same base and it is ending at the same base, right. And if you can see, there are certain deletions in these 2 samples K and A, but in 1 and 2 there are some bases, those are present G, T, A. It means this G, T, A is actually insertion of bases in these particular sample. So, these are my sequence based variations only, these are not errors, correct. So, I had shown you these samples because here you can see the variability also and the deletion is also there. Then this variability, in some it is T, another it is A. If you will further go up, here it is G, A, these 2 bases are insert over here, the base insertion is there, here also the insertion of stretch is there, here also the insertion of stretch is there, right. So, this is how we have prepared our file for GenBank submission. So, this is the nucleotide form of that file. Okay. Now, should I move further, or you have any query? You can point it out right now only, otherwise you will get confused. Yeah. Right.
Actually, I don't know what that consensus sequence means. It is similar or not?
Consensus is, yeah, contig is like what contig is. Consensus is actually similar, right? Contig is that you are joining the region considering that overlapping region. Consensus is similar among all. Okay. If, okay, now this is the region, right? Now my fingers are consensus, but the whole sequence is contig. From here to here it is contig, and this is consensus. Consensus is actually the similar, the conserved among all, right? But contig is the two sequences you are joining considering that overlapping region as a single one. Either you will consider this, or it will cumulatively it will form it as a one.
That means we don't need to do any consensus sequence then.
No, you cannot make, how come consensus, here you can say that consensus, why do you require? Since you have two sequences, you want to join those two sequences, so that is the contig formation. That is a contig formation. Okay, that is a different one. Huh. No, when you will be making contig, na, the software will take care, it will already convert it to reverse complement, and then it will do. But when you will be checking it, let's suppose in forward you cannot find that good base, right, and you want to cross verify it in your reverse sequence, that particular sequence, like chromatogram file of your reverse primer, that you have to make it reverse complement, and then if you will check, then you will find it, right. So how you can do that? If you will go to Finch TV, this is the Finch TV, go to view, okay, then here one option is there, reverse complement. If I will do this, then the sequence will be converted to reverse complement, right. Now it is reverse. So for the reverse primer, you have to check it in the reverse complement of that particular sequence, correct. So now after completing this particular exercise, you can export these edited files to make one document for that. So how you can do this aligned file, there is one option, select this file, go to file, export. What will happen? Your trimmed and edited version will be exported to your text version, right, that you can use it further for your downstream process. So there is one option, FASTA alignment, open, next, just to show you, I am downloading it to desktop only. So here that file has been exported, right? The edited version will be exported for you. So all those edited files you can find it over here, right. You can save this document, because you will require this document while submitting the sequence in any of the database. Now you can make all these things, like suppose you are handling 5-6 genes at a time, you can do this exercise for all those genes, keep the files ready as the gene name, the nucleotide format, right. Now you have to convert these nucleotides to the protein form also. Okay. So what you will be doing, you have to make use of one software is there, XSpeci translate tool. Here also you can make in CLC also, you can convert the nucleotide sequence to that protein version, right. You can see this option is there, na? I had shown you. Translate to protein. So with this format also you can translate into a protein sequence, correct. So maybe further discussion we can have after tea. Okay. Likewise, like I had shown you that nucleotide file, similar way this protein file also, for each sample, sample number 1, 2, 3, 4, 5 and the primer name I had kept and everything I have pasted here, right. So, what you have to do, there is also some editing required over there. At some places where the initial that codon is there, M-methionine will be there, right. So, methionine, the whole word you have to replace with the code M, okay, with the code M, that it will specify while you will be editing it. I will just show you how it will give you the hit. See here different versions are there, the compact methionine to M and stop codon, it will highlight as dash, which you have to delete from your sequence before preparing the file for GenBank submission, because wherever the stop codon will be there will be a dash, okay. Wherever the methionine will be there is MET, that MET you have to replace with M, and that stop codon, that hyphen type of thing will be there, that you have to delete before sending it for submission. So, this is one file I have prepared, you can just have a look, correct. So, it means in this whole sequence from here to here, this region is coding, from this to this again some intronic region is there, from this region to this region again the coding sequence is there, from here to here again the coding sequence is there. So, what will happen when you will be putting this thing in the preparation of flat files, you have to mention the coordinates of the coding region. If you will see some sequence in NCBI, I think I can just show you how it looks like. If you will just see in the coordinates and the complements, it will show you some figures from like 1 to 10, then again that greater than equal to sign, then again I will just show you how it looks, right. So, this protein file also should be ready before that flat file preparation. Now, you have two things in your hand, one is the nucleotide file and another is this protein file, right. Now, we will start with how to make the flat file. For this thing, Sequin is one of the version that is in NCBI only. So, download that Sequin, that also you can use it later offline, right. So, you can make, how you can make all samples with one gene, you can put it in a single file, right. All samples like 10-20 samples are there with a single gene with the same region amplified, you can annotate and put it in a single file, and every information you can add on to that format, it will generate a flat file. That flat file you will have to send through some version is there, Sequin macro send. So, you will be sending it, putting your information, they will ask you your name, author name or your email ID, all this information that will ask you and that will be submitted. So, I will just show you what is the Sequin. So, here there are 2-3 options. One is the start new submission, right. Another is the read existing record. I will just show you from the existing record how it look like. but once you will be starting the new thing, you have to go with the start new submission, because you have to fill down the information of the author, your institute name, your fax number, your title of the job, what all are the authors, what is the study regarding. So, these all information you have to put stepwise. So, right now first I will show you how to read the existing. I am already having one flat file ready with me. I will show you how it looks like, right. So, go to this read existing record. See, this is the flat file, this is what we call flat file. So, here stepwise we have added all the things like the organism name, this was the sample name, right, LB1. This was my first sample with this primer. You can see at the top, this is LB1 with PGK. If I just scroll down, other samples are also there, but in my window I can see the sample one by one. So, I have to select it from the top and then keep on checking my all the entries. So, now it will highlight that how much bases are ready with you for submission, right. This is the date, this is the organism name, these all are the authors, this is the title of the study, right. Then how many bases that it will recognize itself, then what was the technology you have used, it was the Senger's technology. Then you go down, then this is regarding again whatever information I was telling, that you keep it ready in some document and keep on copy pasting it. You do not have to waste time, like at the spot you cannot do everything, you keep the things ready and keep on copy pasting it. So, this is the genomic DNA, this was collected from the leaf, this is the country, collection date, collected by, identified by, and this was the CDS region I was telling you, that what all are the complements that you will join that will make the protein region. So, where I will specify that if I join the coding sequence from nucleotide number 2 to this to this and this to this, that will make that protein sequence, the coding sequence. So, this is the continuity of the coding sequence. In the nucleotide, it was fragmented into that coding, non-coding, coding, non-coding, but here those people, they want that you have to specify the coding sequence along with the coordinates, then only they will accept it. This is for all type of genes, whatever gene sequences we are submitting. Although the journal like the identification, bacterial identification and this fungal identification, there we do not have to specify because that is the universal marker only. But if you are going with the gene wise, then you have to specify this thing, right. So, once you will specify that thing, what will happen? All the stop codons that will be eliminated from the sequence, right. In nucleotide, you had seen the some stop codons are also coming in between, because wherever your coding sequence will end, there you will get one stop codon, right. So, here that thing has been taken care, and not a single stop codon is visible over here, since we have mentioned the coordinates of the coding sequence only. So, it will keep on asking you what all formats you have, and it will keep on highlighting in this format, this is the protein sequence and this is the nucleotide sequence. So, this is actually the visual of whatever, whenever we search something in NCBI, we see this format only, right. You people must have tried only that whenever you are finding any sequence or searching any sequence from NCBI, you will get the format of this thing only, because whatever I am entering for my sample, that only you can see, right. So, this is the flat file. Okay. Now, I will just give a glance of like what all are the steps you have to enter. Now, if I will change this, let us suppose this was for sample number 1, if I will change this, this will be sample number 2, So once your file is ready, let's suppose you want to make some editing in this file. Suppose you missed something, or suppose you have wrongly entered anything. So what you can do, you can directly click on, like suppose I need to change that author name. I can double click on that, then the option will come, like whatever I want to change, I can change it over here, and then I can save it. So this can happen with each and every step. Wherever you want to do the editing, you can do it. Later on also, you first make a raw file, raw flat file, then keep on adding or deleting the things, whatever is required and not required. So if I'll just show you how it works. That was one title thing I had shown you. This is the source regarding the organism, right? Like from where you have collected what is the, it will give you several options, like regarding the organism, you can put whatever name of your organism, location, genetic code, then modifier also, there are several options, the country where you have selected, if you just drop down list, you can see there are several options. So whatever is applicable to your sample, you can enter as much information as you want to enter, right? You have to simply click and keep on adding the information in this and keep on accepting or saving that thing. Once you will accept it, that will be directly highlighted in your flat files, right? So if you can just see, there are several options starting from your culture, collection date, country, then environmental sample, genotype, all possible informations you can enter over here, right? So this is another thing. Then if you go to this one, CDS one, here also it will show you several things. The protein file also it will show you. Then it will be asking you the reading frames. Right now we had seen that there are 6 different frames were there. 3 frames were from 5 prime to 3 prime and 3 from 3 prime to 5 prime. So it will ask you which frame you have selected, that you can check from here. If at all the things are not matching, then you can correct it. You can select this region, it will show you what was the name of the protein you have entered. You started with PGK only, now you have to mention that what was that protein, so that all ready-made information is available for the second person who is referring your work. The same thing we are, because if we need to have any information, we just go to NCBI, search everything, we got all the information. So that is the author or the person who did that work that is entering all the information. The more you will enter, the more transparent information will be available for the second researcher, right? So these all options you can highlight and keep on adding the things, right? Here it will show you all the options, whatever things are there, na, that will highlight over here. Any query regarding this? Three frames will be there, na, and 3 will be for 3 prime to 5 prime, right? What's that mean? Okay, 3 nucleotide that code for one protein, right? Amino acid, right? So if it starts from nucleotide 1, the translate will be different. If it is starting from second, it will be different. It will be starting from third, it will be different, and it will go as like, right? So depending on the, because it is a triplet of nucleotide that codes for the amino acid, that's the reason there are 3 frames for each set. 3 from 5 prime to 3 prime and 3 from 3 prime to 5 prime. All possible translates it will be highlighting. That you just study little bit, then that will be clear to you. Right? Any other questions?
We are having 6 frames now.
Right.
Which is the right frame?
Which is the right frame? The first thing is that it should be the maximum coding region is there. right? Stop codons will be less. If you will, what I told you that you do the NCBI blast for the protein blast you did, there you will come to know the maximum identical thing that translate you will be picking. Okay, there is one more option, that is the additional one. I'll just tell you how it works. You go to NCBI and NCBI blast. You while doing the blast, what you will do, you do the blast and there is one option, I'll just show you. Here also one option comes, there you have to select the CDS annotation. If you will select that CDS option, each amino acid will be highlighted on the top of your nucleotide. So it will show you simultaneously. Let it come, then maybe I can show you. Here one option is there, formatting option, right? You go to this CDS feature, correct? Then reformat. Can you just see that it is highlighting the translate of this region on the top of this, right? Then accordingly you can pick from that, you can match the translate from there and you can pick that one. Got it? It is highlighting, see here it is highlighting clearly from this to this is one CDS, right? Then next CDS start from this to this, the next CDS starts from this to this. So the coordinates whatever you are mentioning, that should be correct, otherwise a single base will shift everything to a new thing, right? So you have to very much particular while you will be doing this thing. So you go with this format while you are doing the blast of this nucleotide blast, then select this option for CDS, so that simultaneously you can get this also, and finally you can match and pick the correct translate, and then you can make your document for that. Got it? I think, yeah, you can submit. I haven't done it, but I think you can submit. You can submit. You can submit. Those are the different formats. Information you will be submitting the same only, those are the different formats only, the set of instructions you have to follow, that is a different, right? So any queries regarding that, any confusions? You just try this thing after going back from here, otherwise you will forget each and everything, that I know. It happens, it's not that you will forget, but it will happen with anyone. So once it will be done, then that flat file that is ready for submission, then there is one option, that is sequin macro send. Sequin macro send. Okay, if you will just start na, it will keep on asking you that sequence I told you na, that right now I had told you the existing one only. But if you have to submit it new, you have to follow the stepwise instructions. Now it will ask you like a submission, contact, author detail, affiliation. You keep on adding it, keep on saying next, and it will ask you another instruction. So that is how you have to keep on entering the things. And once, let's suppose you have already entered some information regarding the author, regarding the institute, regarding the title. If you have already made one flat file, then there is one option that those information you do not have to enter it again. There is one option for pick the existing template. So those all information it will directly import from the existing template, and sequence you can add it according to your whatever sample you will be adding, right. So this is how you have to send the sequence to any of the database. If everything is correct as per their instruction, then you will get within 1 week you will get your GenBank accession numbers. Otherwise, if you did any mistake at the nucleotide level, na, if even one nucleotide you missed out, na, then you will keep on struggling for months and months to get your accession number, because they will keep on sending it back that you please annotate it again, check it again, annotate it again. So that curation part in the beginning, you please do it correctly. If you will not do the curation, then the protein translate will not come, and you will not get your entry whatever you have actually targeting the study, the which you are targeting, that you will not fulfill, right. So the curation part, the sequence editing form, that you have to do it very carefully. Base to base thing you have to do. Wherever variation you are finding na, you just check whether that variation is actually the variation, or don't just blindly believe the data whichever the company is giving. You have to actually check each and every base, and that takes the major time. PCR, doing the PCR, send it for sequencing, it's nothing. It's 2-3 hours work only, right. But then to handle those sequences and getting the accession number, that actually matters. So any other confusion, any other question, please? So I assume everything is clear. You just write down these softwares, practice it once, right. From maybe you can start with 1 or 2 gene, pick any sequence online only, try to make this up to flat file, just see wherever you face any problem, then you can get back to us, right. But try it once, and try it today only, otherwise you will forget, because there are multiple softwares and multiple steps are there. If you have not written it stepwise, then you will stuck it at some other place. That only I told you na, that sequin macro send, if you will Google out na, I'll just see if it is coming. It is nothing, that whole file you have to attach along with your information, like the sender's information. They will ask the name, institute, phone number, email ID, and then that flat file which I had just shown you, that you have to attach it there and you have to send it, that's it. They will, after you have sent, they will give you some reference number through the mail, email ID which you have highlighted over there. Now for further reference, you have to add that submission number only, if they want any query or any question they want to ask. Right? this is that software, right? So this thing you have to do online. You have to fill down all the information, then file you have to attach it over there, and then simply submit. That is the only thing. So I hope the things are clear. If not clear, then you can please ask me.
Any question? Any doubt?
Regions has written nucleotide 1, nucleotide 230.
Mhm.
We have to type it manually.
That we have to type it manually only. Like I had just shown you, na, this thing, this part, this I had shown you, na. So now you have to actually manually count it, because it will not be highlighted anywhere. So you have to count it, like from which base it is starting up to which base. That you have to highlight, and then you have to cross verify your translate whether that translate is same, because if you will be shifting even a single nucleotide, then the whole frame will be shifted. So that is actually the extra work that takes lot of time, because base to base you have to match, and then you have to take out this particular translate, right?
Any more doubts? Any questions? It's clear to all of you, or
Can we practice that maybe
no? If you did not understand something, this is an opportunity, right? If you want to clarify anything, because I was watching sitting in my chamber only. I think this was explained from the very basics, right? I would say the sequence analysis simplified, right? So I can make two interpretations: either you fully understood or you didn't get anything out of it. So questions or many indications of that. Anybody is having that, please. Identify yourself when you ask questions.
Thank you, ma'am. Myself, Neelam Washington. I'm doing here PhD third year. So my question is, we all know that the NCBI is having a lot of large database and it's having so many datas, and there's a lot of junk files out here. So because it's so like it's very easy to put any file in this data. So how will we correctly find when we are doing any like comparative analysis, taking sequences for a phylogenetic tree or anything like that, for viruses, any sequence. So how will we know that this sequence is coming from anywhere it is, how much correct it is, or how much good the sequence is?
No, that while submitting the sequence, you know that how honest you are in curating your sequence and send, because it's no one but we only, those who are sending the sequences, right? So how honest you are in curating the sequence, that's the reason earlier they were not asking for annotation and curation, right? Earlier whosoever can want, they can submit the sequence like that only. But now, until and unless they verify that annotation and the CDS, they don't consider it. That is one of the reason that they want the quality check. Whosoever now cannot send the sequence as such.
Yeah.
Maybe the earlier entries must be there, but right now we faced this thing several times, that if any problem is there at any point, they will not give you accession number, then how those entries will go there?
Yeah, the databases are populated by all of us, correct? And there is a saying, sequence analysis, sequence generation from an organism whose phenotype is not well characterized. Technically, you cannot upload that data in GenBank, right? When you do the flat file making, what name you will give, genus name and species name if it is a cellular organism. Maybe it's true with viruses also. You have to give the correct phenotypic information in the data sheet, right? These two should match. Why? Because your mistakes will amplify with the time and space. Let us not forget. So here comes the quality checks nowadays. unless and until you get the correct protein of the gene what you sequenced, GenBank is not going to give you accession number. It's very clear. So, but all said and done, with due respect to the people who populated it, now there is a kind of feeling GenBank is having a lot of wrong and not so authenticated sequence information. For this reason, there are specialty databases have come now, right? In one specialty database for bacteria it is there, that is called PAMDB, Plant Associated Microbe Database. The beauty of this database, again the databases are managed by people like us only. They have to take out their time to maintain those databases. In those databases, the highlight of those databases are, in the case of PAMDB, because I really populated a lot of data there, what they ask, not your sec file or FASTA file. They ask your chromatographic file. Because chromatographic file will not lie, it is the real file coming from the machines, right? And the curator will spend time on those, the chromatographic file, and he will, okay, if you are telling that, okay, I have a gene sequence that is a different allele as compared to the one which is already there, you are claiming for the new allele for the sequence, then your claims are verified by the curator of those databases, then only he is going to accept it, and then only he is going to accept your claim, right? If 100% match is there, then no problems, right? No complaints. If you are claiming for novelty, then the problem comes, right? And how authentic your data, whether it is a base call or it is a sequence error. So this can be deciphered only with the help of the chromatographic file what Dr. Neelam highlighted, right? So purpose of this training is that only. You should be able to read chromatographic file, not the sec file. The Weiser would insist for trace files or chromatographic file, not the sec file. You extract the sequence from the best base calls, right? So again, she made it very clear. I was listening. Be transparent, give as much information as possible while making the data sheet of your sequences. So that makes your, your data referred by other people. They will be using your data in their analysis. So it is the genomic science is all about involving other people who is working in the similar lines. And then only the databases which is in public domain is of any use for anybody, right? Any more questions by anybody? And the students who are sitting in UAS Bangalore and Kilikulam also can ask questions. I'm not sure still they are attending the classes because the sound was an issue again. Anybody? Backbenchers? If no questions, shall we? Any questions coming to your cell phone? No? Okay, fine. Yeah, it's around 5:15 now, and if no more questions, we will formally close this session. And I thank all the students sitting here and also sitting in UAS Bangalore and Agriculture College Research Institute, Kilikulam. And apologies because there was some issue on sound. We tried our best to fix that problem, and I think partly we were successful. And one of the campuses, they made it very beautifully today by putting some extra speakers. And I'm sure the other campus also would have done it. The Bangalore campus also would have done it. So overall, it was a kind of orientation for all of you to understand how the sequence files need to be handled at your end, right? So I thank Neelam for making this session very lively.